Good evening. Welcome to our social media group, Grow Your Own Science and Maths Like a Superhero. My name is Brent Brown. I did my degree in immunology at UCL in London and a master's in pharmacology at King's College London. I've researched human papillomavirus. I've also done antibody research looking at tyrosine as a marker of oxidative stress. My colleague here is Asghar Ali from Pune School in Pakistan. You're most welcome. Asghar is a botanist, and it's a pleasure for you to be joining me from all the way across the world. Asghar, you're smiling. Talk to me.
Okay. Thank you, sir. Thank you. Today we will. I'm very thankful to you, sir, for giving time. Today we will discuss the bacteriostructure and the life cycle of bacteriophage. So first of all, we take the start uh, from the structure of bacteriophage. As we know that bacteriophage is a type of virus which attack on bacteria. So we will discuss their structure. Bacteriophage is a virus which attack on bacteria and kill them. And they're not here are two conditions. In one condition, they will kill the bacteria, and in another condition, they will not kill the bacteria. They will only reproduce inside the bacterial cell. So the from this, and the structure bacteria have bacteriophage. We will discuss its structure. The structure are made of capsid. The capsid may be icosahedral or polyhedral. Uh, this is the capsid. The capsid is made of capsomere, and inside the capsid is nucleic acid. Bacteriophage consists of DNA. It is a DNA virus. As we know that the virus consists of DNA or RNA, but never both exist in the virus at the same type. In plant viruses, are RNA viruses, but animal viruses are DNA viruses, and also the bacteriophage is a DNA viruses. It has DNA as a genetic material inside their capsid. Now, between the capsid and the tail, there is another part which is called collar, and the tail there is a tube. Around the tube is a sheet, and the tail fiber are used for the attachment. The tail fiber, when the bacteriophage attack on bacteria, the tail fiber only are used for the attachment. When the bacteriophage attack to the bacteria, the first step is called the adsorption step. Inside the base part of the tail have a base plate. The base plate have lysosomal enzyme which are used for the digestion of the wall of bacteria. So it penetrate the wall of the bacteria. So there are two type of life cycle of bacteriophage. One is called the Lytic life cycle, and the other one is called the Lysogenic life cycle. First of all, we will explain the Lytic life cycle. We have to explain the Lytic life cycle. So the Lytic life cycle is also known as master and slave relationship. Before we have discussed that the bacteriophage structure of bacteriophage, the bacteriophage size is 20 to 224 to 24 to 200 nanometer in size. When the bacteriophage virus attack on bacteria, we have discussed that it will either go through the Lytic life cycle, or it will either go to the Lysogenic life cycle. So first is the Lytic life cycle. Lytic life cycle is also known as master and slave relationship. Master and slave relationship. Here the bacteriophage will be a master, but the bacteria will be a slave. In such a condition, the bacteria become died, and the virus replicate only inside the bacteria. The first step, the virus attack on the bacteria. They use their tail fiber for the attachment. They will attach at the receptor site. Here, the site at which the bacteriophage attack with the bacteria is known as receptor site. Uh, this is known as the receptor site, and this step is called attachment step or adsorption. The attachment of bacteriophage with bacteria, it's called adsorption. The first step is the adsorption. The next step will be the penetration. The bacteriophage will penetrate the wall of bacteria. It has enzyme known as lysozymal enzyme. Through this lysozymal enzyme, it will digest the wall of bacteria. They will create a hole inside the wall of bacteria. When they create a hole inside the wall of bacteria, they will inject their DNA through the tube into the bacteria. When they inject their DNA to the bacteria, they will take the control of the metabolic machinery of bacteria. In such a condition, a relation will establish between bacteria and the bacteriophage, and this type of relationship is known as master and slave relationship. The bacteria in such condition will be a slave. Are you getting my points?
Very well explained.
Yeah. And the bacteria will be slave here, and the bacteriophage will be the master here. They will control all the machinery of bacteria, all the metabolic machinery of bacteria, and they will they will synthesize their protein code, and they will replicate their DNA inside the bacteria. When they make their DNA and
Sorry,
they make
Asghar.
their
Can,
protein.
can I just interrupt a minute? So we're talking about cell lysis. So cell lysis just means cell lysis in the very straightforward terms is how a cell splits. So this is what happens when a
What,
virus enters,
come, come,
when a
come.
bacteriophage
I'm
enters.
I'm coming to that point after.
Okay.
Okay, 1 minute. Before lysing, they will prepare their own capsid. They will prepare their tail fiber, and they will prepare replicate their DNA. When they replicate their DNA, and when they form their protein code, after the formation of these things, they will exert pressure on the wall of bacteria, and they will when pages made inside new pages made inside the bacteria, so they will exert the pressure on the bacteria, and the bacteria will die in such conditions. So this type of life cycle is known as a Lytic life cycle. The first stage is the absorption step,
Yeah.
attachment of virus to the hosted surface. Now the entry of viral DNA will enter in the second step. In the third step, replication of viral genome and the protein will occur. And in the last step, assembly of new virus formation, new viral component will form. And at the last step, the host cell will die, and new viruses will release from the bacteria, and the bacteria will die in a such condition. So in lytic life cycle, the bacteria will die, and a relation will establish. Such a relationship is called the master and slave relationship. The bacteria will be the slave, and the virus will be the master. And after that is the lysogenic life cycle. So lysogenic life cycle is known as host and guest relationship. The bacteria is act as a host, and the bacteriophage act as a guest. The bacteria will not die in such condition. They will live as the bacteriophage will live as a guest here. When in the first step is also same as lytic life cycle, the bacteriophage will attack on bacteria, and the first step is the attachment or the adsorption. The bacteriophage will attach through tail fiber at the site which is known as the receptor site. When they attach at the receptor site, they will use their base plate to digest the wall of the bacteria. When they digest the wall of bacteria, they inject their DNA to the bacteria. When they inject the DNA to their to bacteria, they will not take the control of the bacteria here. They will only synthesis. They will only replicate their DNA along with the bacterial DNA. When the bacteria replicate through binary fission. The formation of a new DNA will occur. Bacteria will divide into two daughter bacteria. Here also the viral DNA will replicate, and in such condition they will not die the bacteria. Some part in such condition, some part of bacterial viral DNA will incorporated into the DNA of bacteria, and when bacteria divided into two daughter bacteria. So some part of the virus will also move to the along with the bacterial DNA into the daughter bacteria. So in such condition, the bacteria will not die, and this type of relationship is known as master, so guest and host relationship. So this is the lysogenic life cycle. So we have discussed two type of life cycle here. One is called the Lytic life cycle, and the other one is called the Lysogenic life cycle. And also we have discussed the structure of a Bacteriophage. So this is enough for today lecture. And
Okay.
this is at the last we will discuss the summary of the structure of bacteriophage, uh, uh, Lytic life cycle and lysogenic life cycle. So in structure we have discussed that bacteriophage is a virus that attack on dead parasite on bacteria, and their size is 20 to 200 nanometer, and they are tadpole shape. Their structure is that of tadpole shape. Their the head is also known as capsid, and head may be of different structure. They may be icosahedral or hexagonal or filamentous. Biochemistry, they are made up, head is made of protein. The function of head is they protect the genetic material of the bacteriophage, and the genetic material of the bacteriophage is DNA. It contains only DNA. It will not contain the RNA. And the function of the genetic material is they control all the metabolic activity of the bacteria. The tail function is only the attachment. They are used for the attachment to subpart. Bacteriophage have collar. It is a junction. It is a junction of head and tail, and they have tube. The tube are hollow tube used for the transmission of DNA. Sheet around the tube is a protective covering, which is known as sheet. And at the lower side of the tail is a base plate, which is used for the drilling, which will digest the wall of bacteria. So it's the about summary about the structure of bacteriophage, Lytic life cycle, and lysogenic life cycle. It's enough.
Thank you very much, Asghar. That was very well explained. So I'm going to stop the recording there. Thank you for everyone who's joining this and watching. There's a short section here at the end which is on different virus structures, and we're going to do another section on that next time. So you're most welcome. Thank you very much.